So let's go to the second part of the results. Here we start to couple components. So we will give a periodic stimulation to a component Y1, and this Y1 will activate Y2. We want to know if frequency preference in Y1 could be transmitted. So this level is as before, and then we add a second level here that is a function of Y1, and the important parameter here will be the relative time scale, tau, that is the time scale of component 2 relative to the time scale of component 1. So we need to make some decisions on which are F1 and F2. Suppose, I'll show only one or two. Suppose we combine a concave one with a linear one. What do we have in isolation? That there is a transient frequency preference here and nothing here. A linear function, remember, that was not able to produce a frequency preference. And so what we found is that depending on the relative time scale, we can really transmit the frequency preference to Y2. However, if we combine linear and concave, there's no way to obtain a frequency preference. So this result here is illustrating what I said at the beginning in the introduction, that a fast component may be reading the transients. So even though this component has only a frequency preference in a particular time window, if the downstream component is fast, it will read that frequency preference. Then we complicated things a little bit. We added some feedback in the cascade. So the equations are as before, but we added a term here that because of the interaction of Y1 into Y2, some feedback comes here, and we want to know if this feedback can modulate the response in Y1. Then, just an example, we combine a linear and concave linear for F1, concave for F2. Isolated, there's no frequency preference here. However, because of the feedback, we can get a frequency preference here. And an interesting example is this linear and linear. There's no way to get a frequency preference in either of them, and because of the feedback, you get a frequency preference in both. And finally, because this way of coupling is really a toy model, so we decided to model this in a more detailed way. We have a ligand-receptor reaction that activates the phosphorylation cycle. There is an enzyme doing the activation, another enzyme doing the deactivation. We took parameters from the literature, and this is what we got. There is a frequency preference at this level, the first level, the complex ligand receptor, and also a frequency preference in P, which is at this level. And if we compare what you get in isolation in each of them, there's not really a change in this one, in the complex, because of adding something downstream. However, there is a change here in the phosphorylation, dephosphorylation cycle, because of having this component upstream. Basically, when we combine isolated signaling motifs, what we found is in a cascade, a transient frequency preference is better transmitted if the second component is fast, which makes sense. It needs to capture the transients. The cascade with feedback, a new transient frequency preference can emerge from feedback connections, and a more detailed model, the frequency preference is transmitted and modulated by the pathway. So in the last part, let me tell you a little bit about the analysis of experimental data. This was done in the summer before coming to the conference, so it's still work in progress. But what we are doing is we are analyzing published data, different databases, as the ones you can see here. We need them to have some features. we need them to be capturing the transients and the periodic regimes, as you can see here. It is clearly seen here. We need the measurements to be done at different frequencies of stimulation so that we can reconstruct the output versus frequency response. And very important, we need to understand the signaling so that we select which is the output. In what I showed so far, it was theoretical and computational calculations, so we decided to use the integral in one period, the accumulated integral. Now we need to know which is the relevant output for each case. So the questions we were addressing when I started this part of the project were the following. Do available techniques provide the type of data needed to validate our results? Can we find significant differences in frequency responses when comparing early versus late responses? Does TFP really happens? How do signaling systems make use of it? I would say, even though the analysis is still, we are still doing this, that there is a yes in the three first answers. The other one is more complicated. I'll show you some results. So I selected for the talk these results. This is a very nice article from 2 years ago in Science, where they studied if failure of signal transmission dynamics could be associated with diseases such as cancer. And so this is the study they did. They applied a periodic stimulation in the ERK pathway, activating ERK. They do it both in normal cells, in cancer cells. They can do it for 2 different pulse durations, 10 and 20 minutes, and for 3 different pulse durations, 5 minutes, 10 and 40. And this is what they got. The black traces you are seeing is the mean of several cells. So far we are working with the mean traces. We are in the way of trying to get individual traces. And what they got is the following. Normal cells has fast kinetics, 5 minutes, okay. And by activating in this way ERK, they have no response. However, cancer cells, they have slow kinetics and they induce improper proliferation. They conclude that cancer cells fail to perceive gaps in the signals. If you focus on this response and this response that have a very short gap between signals, seems that the activation, the ERK is always in a high level. It's not able to go back, and that's because its kinetics is slow.
Sorry, do experiments conserve the dose?
No, not this one. Another set we are studying conserves. So they conclude that misinterpretation of dynamic signals induce proliferation in response to otherwise non-proliferative inputs. So what we did was a frequency response analysis on this data. As I said, we analyzed normal traces, cancer traces. Because of having 2 different on durations, 4 different off durations, we can add them to get 6 different periods. The 1 over period is the frequency in 1 over minutes. So this is what we get, a plot with 6 data points. Which is the outputs? That's the important question. In this article, it was stated that the important output is the integral above a threshold. So we computed the integral of the response above a threshold divided by the integral of the stimulation. That's the gain. And so this is what we got. For normal cells, that's the output. These are the 6 frequency data points here. And why different curves? Because we integrated up to 20 minutes, 50, 80, 100, 150. So as I said, I am moving S. And basically, we can say that there's no frequency dependence in the analyzed range, right? So we propagated errors here, that's why the dispersion you see here, and it's more or less constant. This is in total agreement of previous data in which they measure the ERC gain as a function of the frequency. It is a low-pass filter with a bandwidth of about 2 to the minus 3 in hertz, which is 0.12 in 1/minute. So this measurement was done in the flat part of the curve, right? So what about cancer cells?
Can I ask you a question? What is your stimulus?
The stimulation is an optogenetic stimulation.
We are doing that here too. I mean, I know Toucher is doing the optogenetic.
Yes, it's the same author. No, what you say?
What is your this?
No, we are analyzing their data.
Oh, their data.
Yes. And this data is coming from the same group. So what we got for cancer cells. So notice that this is the same scale, and this is the frequency response. So again, measured at different times. So we observe the following. There's apparently a frequency preference around 0.02 here for all the times we analyze, very early times, 20 minutes, up to more than 2 hours. And it's hard to say something about this. Could be that there is a decrease in the response. We cannot say now with the data we have. That could be explained because, as I said, cancer cells have slower kinetics, but it's hard to conclude that from this data. But again, where is the frequency preference? Apparently is in 0.02. What is 0.02 coming from? This data. A stimulation lasting 10 minutes with a gap of 40 minutes. That's this data point. So we are in the process of understanding the implications of this. That is, of course, a noticeable difference from cancer to normal cells. But I have to say that this article has 3 pieces of data. They present the data I showed, so they stimulate this pathway, they measure ERC up to 2 hours with these traces, and this is what we got. Then they measure up to 5 hours the gene expression at 3 different genes, providing this data. We haven't analyzed this data yet, but we did analyze proliferation data. They measure proliferation in response to 19 hours oscillatory input, and this is what they got. This is normal cells, this is cancer cells, and in the vertical axis you have off interval, in the horizontal axis on interval. So let's say you start here and you go down with a vertical line. What you are doing is fixing on, decreasing off. On plus off gives me the period. So going down means decreasing the period, increasing the frequency. So if you do that, you will see that it only decreases following this scale here. The same starting here. You go this way, you are fixing off, decreasing on. So you are decreasing the period, increasing the frequency, no maximum. However, in cancer cells, we can see this. There's a maximum there. You get this maximum going this way or that way. So we computed what we get there, the frequency response at the proliferation assay. Normal cells have no maximum, cancer cells have this maximum at the same frequency we analyze here in early data, 0.02. However, I have to say that this 0.02 is not coming from the same type of data. Here we have short signals with long gaps. Here we have long, strong signals with short gaps. So even though the frequency is the same, we have to understand this in detail. So still a lot to be done.